Hello, everyone. Welcome to MetaCell's second webinar on exploring the brain of a fly. My name is Matteo Cantarelli. I am the CTO of MetaCell, and let me first thank you all for taking the time for joining us. We are pleased to have three special guests from the Virtual Fly Brain project with us today. We have Marta Costa. She has been with the Virtual Fly Brain project since 2011, working on image integration, ontology editing, and data curation. Marta is also the project lead on the Drosophila connectomics project at the Department of Zoology at the University of Cambridge. We have David Osumi-Sutherland. He's one of the founders of the Virtual Fly Brain project. He works on semantic integration of data at the European Bioinformatics Institute for projects that include the Virtual Fly Brain, the Human Cell Atlas, and the Monarch Initiative. We have also Robbie Kurt. He's the lead developer on VFB since 2013. He's based at the University of Edinburgh, and he has a background in neuroinformatics. Let me tell you a little bit about our company. MetaCell is 6 years old. As MetaCells, we are passionate about building software for neuroscience, a lot of which has to do with visualizing and sharing data and models online to help researchers and teams collaborate together. Throughout the webinar, we're going today to have some interactive polls. So we'll be asking you some questions to get a better sense of what interests people in the audience. And you will also have the opportunity to ask questions throughout the webinar using the software at any time. You can just type the questions using the little software that should be appearing on your screen if you join this, and we'll answer the questions at the end of the presentations. So as MetaCell, we work with a large variety of organizations that span from universities to pharmas, small companies, and non-profits. And let me start this by having a first poll to understand the breakout of the room in terms of who works on academia, industry, and so on. So you should have a poll appearing on your screen, and now you can go and pick one of the three options. I will give some 10 seconds to catch a breath, and I see numbers are changing. Okay, it looks like, yes, they're still changing. I won't stop it until I see that the system is stable. Okay, looks like we got some results. So this is the breakdown of the room today with us. So we have 75% from academia, 10% from the industry, and 15% others. Thanks everyone for answering this. I think it's useful in general for everyone to know what the audience is. And so let me move forward introducing Virtual Fly Brain and how it can be used to explore the brain of a fly. So VFB does extensive curation of literature in collaboration with Flybase and allows users to explore diverse types of annotated imaging data, from confocal slices to neuronal reconstructions. VFB is a joint effort of different universities and research groups. We have the European Bioinformatics Institute, the University of Cambridge, University of Edinburgh, the Medical Research Council at Cambridge, and it is funded by the Wellcome Trust until 2021. Okay. As MetaCell, we are working together with Virtual Fly Brain to develop the version 2 of the web applications that enables integrating an even bigger variety of imaging data. And I will move to the speakers right away now with Marta, David, and Rob to answer some questions and explain us how VFB works. But before we start with that, let me quickly launch another poll. As you gathered, VFB is a hub for Drosophila melanogaster data, but many of the solutions we will be discussing here today are reusable also for other model organisms. So what I would like to ask quickly is what model organisms do people in the audience work with? So you should be seeing another poll. And I will leave it again some 15 seconds. Thanks, everyone. It's fun to see how the numbers are changing as you're voting. I know who's winning, not surprisingly. Okay, looks like we have something that is probably final. All right, let me share the results. So we have 58% of the audience working with Drosophila. Again, not surprisingly, 11 work with the mouse. We have 16% of C. elegans aficionados, 5% with rats, and 32% other that probably don't work with any of this. So thanks for this, and I would move on then to Marta Costa for our first question. Marta, and I think it's the main thing that everybody is wondering, what different type of imaging data are you integrating with Virtual Fly Brain today?
So I guess, so there's two main types of image data. We have a lot of light level data, so this would be confocal, and this is, and we already have a lot of this on the site, and this is because there's been a lot of work in Drosophila over many years in terms of developing these tools and developing libraries of light level tools that can be used. And so this might be collections of single neurons, and so one neuron labeled in a brain. And this is very useful in terms of classifying and characterizing neuron types. And so being able to give a neuron type to a single neuron within the 100,000 neurons of the adult fly brain. And there might be subsets of neurons that are labeled. And these might be sort of just genetic driver lines, so for expression patterns, or cells that are connected by lineage, so lineage clones, for example. And these types of genetic driver lines allow us to kind of target and manipulate specific neurons, and so testing their function and seeing what they do in a circuit. And these are types of tools that researchers are very keen to search and to be able to search for, and to be able to actually pick the right one for their experiments. And so VFB has a lot of these data sets that have been generated for the most part by large data producers. In terms of connectomics data, this is something that we're heavily focusing on sort of the next few years, that the data is starting to come out. And so part of the work, for example, that I'm involved with Drosophila Connectomics Group, working on generating a connectome for the adult brain, we're using electron microscopy data. And this allows us to not just reconstruct the neuron, but get a synaptic resolution of every synapse of that neuron. And so this is, and one neuron might have hundreds to thousands of synapses. So this is actually, so why we kind of trying to now develop new tools to be able to deal and to display and to generate ways for users to search this type of data, which will be very data rich, but being able to do it in a way that's kind of user friendly. And so for the most part, VFB at the moment has a lot of the light level data. So people are able to look at genetic driver lines and use the tools on the site to be able to pick which one might be most useful for their experiments. And the electron microscopy data, so the connectomic data, is coming.
I see. That's great. And how are you acquiring and processing the data?
So for the most part, so we don't acquire the data ourselves in terms of generating the confocal stacks. So we have relationships with large data producers, and they would be sharing the data. But so once we have the data, and or in collaboration with these data producers, the main thing is to be able to align the image data onto a standard coordinate space. And so this is what we'd call a brain template. So being able to take different data sets that are generated in different labs, which means that the brain that you're using is slightly different, and taking that data and transform it to this standard and common coordinate space. And that means, we use this transforming techniques. They're called image registration, and there are many different ways to do it. But the main thing, it means that that we can overlay images from different data sets and compare them very easily. And so users on the site can just take an image from an expression pattern and a single neuron, which are coming from different brain templates and different experiments, and see them on the viewer at the same time. And so we can use image registration to register onto a standard brain template, but we can also use it to create ways to transform data from different brain templates into one. So bridging, sort of kind of hopping across from different templates onto the one that we use in VFB. And these are techniques that are now quite established, and we can do that quite easily. We've also, yeah.
How long does it take to do the registration?
Taking an unregistered image, so a raw image, it might take, let's say, a couple of hours. But bridging, so taking an image that's been registered to one brain template and then bridging it to the one used by VFB, it's very, very quick. And so we'd usually, so when large data sets are formed, usually people will create their own brain template and register it to that. And then we just need to generate a bridging registration to our own. And that makes it quite easy to transform data across. We also used kind of similar techniques to be able to bridge EM data, so from the connectomics efforts onto the light level data that we use in VFB. That means that we can actually trace a neuron in the EM and then overlay it with any expression pattern or single neurons that have been obtained in the light level data sets. And I'll just quickly also say that, so in addition to kind of registering the images, we can also automatically, we have ways to automatically calculate the overlap of each image onto each neuropile in the brain. And this is because the Brain Name Working Group in 2014 designed or sort of defined a nomenclature and sort of the 3D borders of each neuropile in the adult brain. And so we can take that parcellation and just ask very easily within this image and within these voxels which ones fall on neuropile A or neuropile B. And so and that data can then be used to get queries to say, give me all the neurons that overlap, for example, the antennal lobe.
I see. And you mentioned the fact that you're not directly acquiring the data, but rather you're working with other groups that do so. Then what is the process for submission of this data? Say, if you are a lab and you have some Drosophila data that you would like to contribute to VFB, how do you go about doing that?
So, so there's slightly different procedures. So for large data producers, we already have established relationships. And so, we work together and we collaborate in terms of sharing the data in bulk and sharing metadata associated with that. For kind of small data sets that might be published with a paper, we've had actually cases in which authors have approached us wanting to share their data via VFB, or we've approached authors because we think their data set is very valuable to have on the site. And so for for kind of the small data sets, what's most important is that the data is registered, or that the data can be registered. And to be able to be registered, one of the most important things is to have data of a high quality. And so there's, so you can see there's a link on there, which if you go onto that page, onto the contribute page, onto the VFB site, you'll be able to see there are links to how to stain brains so that you can get a clean image, so no background, and how to register the images. So there's, for example, plugins that can be used, or how to bridge data if you have it in one template and you want to put it onto the template that VFB uses. But like I said, we can always work with the authors to help them if they have never registered images, for example, or to offer advice to look at their data set and see, do we think this, we can register this data set, or what changes might need to be done to be able to do that.
Okay, sounds great. And before moving on, some questions for David, but before moving on, we've seen from Marta different imaging types that are integrated by VFB, and I have another poll that I would like to run. So if people could fill in what, answering the question, what type of data would you be interested in visualizing? So we have light level data, electron microscopy, electrophysiology, reconstructed surface models, and connectomics. Thanks everyone that has voted. There's still some that haven't yet. Okay, I think we reached. There was an extra vote that came in. Thank you, everyone. So let me share the results. We have a sort of quasi-uniform breakdown with 59 on light level data, about 30% on electron microscopy, about 50% on electrophysiology, reconstructed surface model at 35, and connectomics at 53. Thank you for this. Is all going to be very useful. And so, David, we've now with Marta, we've seen, we looked at the different type of imaging data that VFB is integrated. How does VFB add a semantic layer over this?
Yeah, so we integrate very diverse types of data. So as Marta's just described, one type of integration is image-based, where we register everything into a common image space. But that'll only get us so far because we have lots of information and data that is not entirely image-based. So working from lots of different data sources, so annotated image data, working on annotated connectomics data and these different parcellation schemes. So we've got one coming in for the larval brain as well as for the adult brain, and working with Flybase to curate from the literature. We have a couple of technologies we use to integrate this data. One, and I'll just very briefly mention. So we use graph databases and a language called OWL, and this gives us a way to this gives us a mechanism to sort of make a unified semantic integration of lots of different data types. So part of our job is devising standard identifiers and descriptions for entities like cell types, so a projection neuron, or transgenes that you might use as a driver to target expression, and then the relationships between them. So the standard way we have by having standard semantics and standard ways to express these, and extra sort of logical tools for reasoning about them, we can integrate very different types of data and have a pretty simple standard unified query language to query across it. So the types of data we collect, so working with Flybase, we capture information about genetic features, transgenes, for example. We curate detailed descriptions of neuron types and record information about their properties. So that might be the neurotransmitter, some general information about their function. So maybe their the sensory modality they're involved in, and then where their synaptic terminals are, what neurons types they connect to. And then we also use the same kind of semantics to curate images. So we curate images of expression patterns or single cells, and we're working on a system for importing information about so raw connectomics data as well as reconstructions from the CatMade EM database. So, I mean, with those standard identifiers, they're potentially usable as well for any other users to annotate their data and make it easy to integrate with us or potentially with other users. So Flybase identifiers or the identifiers that we have for cell types, for example, neuron types. We're building systems so that within the next year or two, we should have easier tools for users to curate their own data within using external spreadsheet type tools and also within VFB.
Okay, and how does the semantic integration fit with what we were looking at with Marta earlier, with the image-based integration?
Actually, Marta is lead author on a paper with Greg Jeffries on a blast type system for neurons. So this actually works by when neurons are registered into the same space, you can take a skeleton of one neuron and use that to search for neurons with similar location and morphology, and that gives back a ranked list of hits. Now, actually, if you could move on to the next slide, that has enabled us to, so NBLAST itself, when you do a blast of all the tens of thousands of neurons that we have in our database, this identifies known neuron types and predicts new ones. So if you do make a big blast matrix cluster, use an unsupervised clustering, then out of that fall a lot of known cell types like this alpha beta Kenyan cell, as well as lots of new predicted cell types. And that's allowed us to, that's made the job of annotating our single neurons much easier than it otherwise would be. You don't have to go through and look at tens of thousands of cells. You can look at single clusters and annotate those. Now, so that means that we now have illustrations, 3D illustrations of many of the types of neurons that we have on VFB. And one of the powerful things we can then do with it, if you move on to the next slide, is that so NBLAST can actually be used when you have an image for a neuron. It can be used quite effectively to search for candidate transgene expression patterns, so transgenes that can be used to drive expression in cells with this morphology. So given that we already have once we already have a mapping from cell types, neuron types to these images can then be put into this BLAST system, and we can use it to search for candidate driver lines. Now, the NBLAST in this context is not 100% successful, but it gives a pretty reasonable, it gives a hit rate high enough that people can go in and actually look for themselves and check using our 3D viewers whether by eye it looks like these things line up as well as according to the NBLAST queries. So I think that really just shows the power of being able to integrate this imaging-based integration techniques and tools and queries with the semantic integration.
Okay, and what tools can be used to make it easier, David, to navigate through all of this data that you're putting together?
Yeah, I think, this is clearly we're integrating lots of different information. So I haven't mentioned we also bring in phenotypes as well, and we're going to have an increasing focus on behavioral phenotypes as well as and bulk behavioral phenotype data, as well as integrating all this connectomic data. So there are challenges in when we have such a rich semantics in presenting this in clear and clean ways. We're developing query tools and browsing tools which give graphical representations of the semantic information we have. So this is an example of one we're working on for connectomics data, where you can query for all the shortest path connections through connectomic data from one neuron, single neuron type to another. So in this case, from this ipsiphone neuron in the brain to this sensory neuron in red. And we can layer over onto this all of the semantics. So we know that this is a sensory neuron because of the semantics. So that's layered on as an annotation in color. And through this gives, I think, a way to very rapidly go between connectomics data, information about drivers, images of neurons, expression data, etc. Okay, so I think that kind of summarizes where we're going with that.
Oh, okay. And a quick question for Rob. Rob, how do you configure then the VFB system in order to be able to query this data?
Okay. Well, one of the things that we kept very much in mind when we set up the system was to make it as flexible as possible, because the systems we use in the background we may change in future because we find better ones that off the shelf that do that. So what we've done is configured the system to be able to talk to any standard API, and we've configured a system to set up queries in a very easy and declarative way. So we just set up, for example, if we want to know, in this case, any presynaptic terminals in a particular area, then we just configure a name for the query. We specify what it applies to. So a query, if you're looking for presynaptic terminals, this is only going to apply in a class of neuropil.
Rob, sorry to interrupt you for a second. Could you speak up a little bit? The volume is a bit, I don't know if the microphone moved or anything. The volume is a bit.
How's that?
Much better. Thank you.
Sorry, it's probably turned around. Yeah, so basically we can apply where these queries appear, because for each different part of anatomy, you'll want a whole different set of queries that make sense. So we can easily configure that when we're setting up the query, and then we can combine queries from multiple APIs. So we're running an OWL query that brings back all the presynaptic terminals in a particular area, and this is quite a complex thing that requires reasoning to know that a subpart is part of the thing further up the chain. And so that does a lot of complex stuff to give us the set of classes, and then we can go through our Neo4j database and look for any subclasses of those particular classes that happen to have images attached. So we can then pull back everything. And so we can combine the best of each of the APIs to give us what to the user looks like a very simple query, but is actually doing a lot of complex stuff in the back end.
Yeah, there's definitely a lot of complex stuff by the look of it. And so, Rob, we've started with Marta looking at how the data is registered together, processed. David has shown us how the data can be augmented and annotated with semantics. And you have this rather complex querying system. What can you do once you put everything together? And I will, do you want to share something? I will. Okay, let me.
Okay.
All right.
Because I can't look at the time.
But keep the microphone close.
How's that?
Better.
Right, basically, I just wanted to show here the fundamental thing that VFB started with is demonstrating anatomy and making that accessible to people who haven't spent 15 years studying anatomy, to know where they are in the brain in 3D. We've previously done this using just 2D slices, which enabled you to move through the stack. And what we've been able to do here is now add 3D context to that. So you can see precisely in the 2D slice where you are in 3D context. And also as you zoom in, you know precisely where you are in that 3D context. Also, we've been able to improve the 2D slice viewer so that you now load the entire stack. You can now move through it very quickly. We've also got 3D volume rendering as well as skeletal rendering. As you move across the 2D slice, you're able to select any particular area and see what particular class of anatomy that is. This enables you to then add that to the display in both the 3D and 2D. As you see, we've also support full orthogonal views. We still have a search, which is much improved on this version. So we can search through any name, any synonym. So if you just find a name in a paper, you can find what that refers to and all the references for it. You can add anatomy by simply clicking on any of the thumbnails or by clicking on the 2D or the 3D. They're all synchronized up. So that enables you to build up a picture showing any particular anatomy you're interested in. And I think I'm about to go. Yeah. So basically from simple anatomy is good enough, but when you actually want to start getting into our massive array of neurons, the easiest thing to do is most people are interested in particular neurons that go from a place to a place. So for example, if we want to go to the procerebral bridge, which is the little wiggly thing on the back of the brain, which I shall show you there. And say we were interested in neurons that go from there to, say we don't even know what it's called. Say we want that little thing in the middle there. Just zoom back. I'm just showing you on the 2D slice. You can go through and look at anything. So you just click on there. You don't even need to know what it's called. Just add that. So we can see that's the nodulus. So if we just get rid of the main brain to make that clearer, zoom in there. If you don't know where you are at any time, you can click on the little plus button with the magnifier, which will zoom you in on any feature. Then we can start building up. You'll see all these queues here. You've got sets of pre-rolled queries. So we want the inputs from the postsynaptic bridge, so that's postsynaptic terminals. That we have 102 results. These are classes of neurons. So each of a class may have images, it may not have images, but most of these have multiple example images from that class of neuron. And obviously with the classes, you can just flick through a number of examples or open the class itself. Now we're interested in ones that go from here to the other one. So we don't want to have to sit here and look through all of them. So what we can actually do is in the query builder, you can then specify a second query to limit that down. So we were interested in the nodulus, and we're interested in outputs ending there. So that's presynaptic. And then that brings us down to 41 classes of neurons. So that makes it a lot easier to start looking through these things. So then again, we can actually select one of those classes. And what I want to show you here is the fact that if you look at the class, where it in the relationships tab we have has presynaptic terminals in, but it's not actually in the procerebral bridge. It's actually a particular slice of it. So we're actually, a lot of our detail is quite fine-grained. We may not have painted domains for it, but you can still run queries from there. So we've selected one of those neurons by clicking on it. because it's already been clustered by MBLAST, we can look for any similar neurons to this one. So if we just select, there's actually only 2 in this cluster. But if you look at it, I just wanted to show you that what an MBLAST cluster looks like. So again, if we zoom into it, you'll see that those are, MBLAST actually works. They are actually a cluster. It's worth pointing out here, these are actually skeleton reconstructions, which were generated as part of the fly circuit. Whether or not we have reconstructions, that's not a problem, because we can actually display the original raw data. So if we turn off the, you have choices to turn off the skeleton, we can turn on the actual original data, which is the expression data used to identify that neuron. So they're actually points of light displayed on here, which is what you're seeing in the 2D slice viewer. And a lot of these, you'll see the reason why it's quite important to have 3D views on neurons in the fact that in the slice viewer, it's just a single point, whereas in 3D, you can see the full structure. So in the old days, you just had to slide back and forth to try and get an idea of the morphology, whereas now you can simply select it. And again, we can select any neuron in the 2D slice and change how it's displayed. Here we want to, if I pick a different, if we want to go back to the previous query that we've run, any query, you can pull down a list of all the queries you've run in this session, so we can go back to looking at the combined query previously. If we pick another one that actually has a few more examples off of it, just to demonstrate the fact that, so if we pick that one, that's obviously still going between those two, as that's the query we've run. But what we can actually do is, if we look at similar neurons here, we can pick one that's actually, rather than on the left, it's actually on the right, just to show the fact that MBLAST does the analysis invariant of left-right, so it's actually flipping neurons to do the analysis, which is actually very handy because most morphology is actually symmetrical, so it doesn't actually matter whether it's on the left or the right when people are doing the analysis. So that just shows that we can build these up and put them through, and you can see, obviously, you can see by the fact they actually plunge into the neuropil that these are actually been annotated correctly. What we can also do here is we can click on the cluster, and you can see all the particular details of that cluster, its exemplar member, and also you can click on the references, which takes you to the original analysis page for that cluster, so you can find out why it's allocated to that cluster and its MBLAST score. So we keep records for all of these because these are pre-computed, so that you can access all that. Any of these neurons you can select, and so if we were as we were discussing before, you can find any GAL4 markers. This I recorded earlier when the site wasn't displaying images, but this MBLAST at the moment is running as an external service. We're currently pulling this in-house, so it'll actually be fully integrated into VFB2. So if you do actually go on the site, you can click on the link and it takes you to the current VFB version. But because I want to take it to here, so I've just copied the link across. So this is bringing up a GAL4 that hits that is the #1 target that hits those. As we were saying earlier, that maybe not a perfect match, but previously you would have had to look through several thousand expression patterns to hit a particular target. So what you do is you find one that here it actually does follow the line through. It's actually a bit easier to see in the 2D slice, but I'll go through one in a second live. What you can then do is find multiple to put together, and then you can start developing splits, which is how most labs do this. It's also worth pointing out as it's picked a hideous color to put that on. You can change the color and the contrast and everything to develop the image that you want for that particular thing. And at that point, I think I'm going to flick over to an actual live one. Right, yep, you're on there. So get me live. Probably left this for too long. So for example, I think this is the one David was showing earlier. So it's worth pointing out the 2D slice actually still does have a very good use. In fact, it makes it a lot easier to trace the path of the neuron and the overlap with the expression. It's worth saying this site is going to be released today, and that's the address, which I will copy across.
So v2.virtualflybrain.org.
Yeah.
Okay.
So that's we've got a release going out, I think, later today, but that's available to play with now. And we'll be adding more features. We're currently undergoing extensive user testing and sort of changing features as people suggest things. But that will be out soon. Oops.
Okay. Well, thank you so much, Rob. This was extremely interesting. I want to use one final poll now that you've seen all different areas that we're working on here with Virtual Fly Brain, and if you could vote on what is the one that you're mostly interested in. Okay. Still more votes coming in. There we go. Thank you. So results-wise, we have 50% interested on data alignment and processing, 70% roughly on morphological analysis, 17 on ontology annotations, and 67 on data visualization and exploration. Thank you for your answers. So we are at the end. First of all, I would like to thanks everybody for attending. Now I will go through some questions that we got from the audience, and I will pass the questions to you guys, and you pick who is the most appropriate person to answer them. So we have the first question. I have a bicistronic line for a specific transcription factor, very accurately mirroring its expression. I want to identify the neurons marked. Is there an automated way of doing this? Hence, emblast in reverse, going from transgenic lines to single neuron.
I can take that. Yeah, so no, at the moment, we don't have that. So we have a subset of the driver lines that we have available are processed to be able to do the neuron to transgenic driver line query. But you can't yet do the opposite, just because usually if you have an expression pattern, they're quite busy. And so, which means that you'd get, I know, many hits if you were trying to search for single neurons. Or, I know, if you have a very clean line that you might be able to segment, I know, a few neurons, you could do an NBLAST query, an NBLAST search that way. But for busy lines, I know the number of hits that you'd get, which would be false positives, means that it might not be the best way to do it.
Okay. And David, you want to add something? I saw your lips moving.
Yeah, I mean, not for this particular case, but we do have cases where driver lines are broken down. We've got split images coming, but we also have cases where driver lines are broken down clonally into their components. And each one of those is recorded as a fragment of the expression pattern. So one of the expression queries that are coming, given an expression pattern, show me fragments, and those fragments could be single cells. So that's one way to break things down. I don't think it would actually help with this particular case. But if that expression data were broken down clonally, it would be a better fit for our tools, I think.
Yeah, so I guess, yes, I was just add, so I guess one of the things we didn't really mention is that with the data that comes in that we create from the literature, the annotations to say that this driver line is expressed in this neuron type are curated. And so this would be a query type that doesn't use the image data, but that you might able, if it's a driver line that's already available, it might be able to give some hints if that specific evidence has already been shown.
Yeah. So that could be down to the single neuron level, and there is some annotation of actual expression, regular gene expression patterns, not just driver lines, down at that level as well.
I mean, Marta, is it worth mentioning the painted? If you were interested in a particular section of an expression, you could highlight. You could actually mark that area and run an MBLAST from that. Yeah.
So we should say, yeah. Thanks, Rob. That all of these NBLAST tools and image registration and sort of a lot of the analysis that have been developed for the NBLAST paper, but also other tools in Greg Jeffrey's lab, they're all based in R, and so you're able to do quite more complex queries other than just the sort of the web pages that you've seen. And so you can do queries getting all the neurons in a specific region of the brain other than the neuropals. You can select what region you want, for example. And so I know what's available online. It's a sort of a subsection of what's possible if using the code behind these tools. And that's all available. There's there are links on the VFB sites on the useful links section that will list all of the pages where you can get more information on this.
Okay, thank you, all of you, to answer this question. There's another one asking if the Genelia Research Institute Adult Fry Brain EM set is available on VFB.
I think this should be Marta again.
Yeah.
About
yeah. So Rob, so we're setting up a server to serve that data in terms of the actual EM sections, and so that's almost ready, and we will be displaying some data that will be attached to publications coming out of the Connectomics effort through that server. We don't have any examples yet, but we will be releasing the Connectomics data from the adult brain project through VFB.
And so one paper at a time, I guess, initially at least.
Yeah.
So, as they're published, we'll push it live.
Okay, and another and probably last question, although there is more, but there's only so much time we have here today. I suppose one last question, and it might be still for Marta. How much computing power is needed at the back end when you're doing the registration? From the audience are asking if you need to use clusters or what is the complexity?
Well, yeah, Rob can answer that as well. I'll just say that, so for when data comes to us in raw form, so unregistered, and it's thousands of images, it's certainly helpful to have a computer cluster because it can take days, many days to run the registrations. But for a single registration, it's perfectly possible to run it on a laptop, on a normal laptop. It's just, I know when you're doing bulk data and it's thousands of images, then it's helpful to have some more computing time. But Rob has been doing that in Edinburgh as well.
I mean, if you've got a desktop machine, you're probably talking about 8 hours, somewhere to 4 to 8 hours to do a normal alignment, depending on how much it has to adjust. But it's doable. I mean, basically, if you want to leave it overnight, you can do it on desktop machines.
Yeah, sounds like fun. And as well, very final question for David. We looked at all these different techniques that we're using for VFB in the context of the Drosophila, also in relation to the semantics, annotations, and so on. How much of these is transferable, reusable, let's say, for other model organisms?
Well, so we have a stripped down version of the configuration. So the nice thing about the way Geppetto works is that there's a separate declarative configuration where you can declare the basic types and queries that you run. And a stripped down version of that, I mean, would allow for any data set where you have ontology annotations attached to 3D registered image data. It's pretty straightforward to use our pipeline to generate an instance of BFB Geppetto that runs that data. So we're actively looking at trying out other data types on that over the next few months. Data from other sources, I should say. And it doesn't, we have a more sophisticated semantics for dealing with neurobiology queries. The ontology doesn't, and the annotations don't necessarily have to support that, but they can, in that we use the semantics we use is standardized through the relations ontology and a bunch of external standardization efforts that are external to VFB. So it's, the technology stack's not too difficult to work with, and in its stripped down form, it's pretty straightforward to run a generic instance with other data types, with other data.
Okay, sounds good. Let me bring this webinar to a conclusion here. Let me, first of all, thanks everyone for attending today. As you will close, probably you will get a super short survey that it would be very useful if you could take a minute to fill it in. It's essentially a way for us to learn more about you. I would like to thank our guests here for doing a wonderful job presenting a virtual flight brain. Marta, David, and Rob, thank you so much for sharing with us today. I'm sure everybody found the virtual flight brain project extremely fascinating. And for those of you that are in the London area, MetaCell will be presenting at the HPP Innovation Expo next Friday, the 23rd of March, at the Science Museum. So if you happen to be in the London area, come to say hi. We'll be there showing VFB and more of the projects we've been working on. Again, thanks everyone for attending. I hope you found this useful, and I hope I'll see you at our next webinar. Have a great day. Bye.